So today we will be studying about the Novo Purine and Pyrimidine synthesis. In pyrimidine synthesis, we make cytosine, thiamine, and uracil. In pyrimidine synthesis, in we make adenine and thiamine in purine synthesis. First, we talk about pyrimidine synthesis. This pathway builds the pyrimidine ring first, then attaches it to sugar. The starting ingredient it began with pyrimidine synthesis. It began with glutamine plus carbon dioxide plus ATP. Glutamine plus carbon dioxide plus ATP. The enzyme CPS-II carbamyl phosphate synthetase to combine these ingredients to make a carbamyl phosphate. These ingredients are combined together by enzyme CPS-II. CPS-II carbamyl phosphate synthetase 2, and then it make carbamyl phosphate. Okay, glutamine plus carbon dioxide plus ATP. These are combined with the help of enzyme CPS-II carbamyl phosphate synthetase to form carbamyl phosphate. Now we will discuss about CPS1 and CPS2. CPS1 and CPS2. CPS2, it is in cytosol. Cytosol or cytosol. It takes place in cytosol. CPS1, it takes place in mitochondria. So how we will remember this? Mitochondria. Okay. Now we will talk about another step. Aspartate is added to carbamyl phosphate to create orotic acid. Carbamyl phosphate is made. So aspartate is added to carbamyl phosphate to make orotic acid. Carbamyl phosphate plus aspartate. It is used to make orotic acid. Leflunomide inhibits the enzyme dihydroorotate dehydrogenase here. It is used to treat rheumatoid arthritis by stopping white blood cell replication. So we will remember a drug called leflunomide. This drug block inhibit enzyme dihydroorotate dehydrogenase to treat rheumatoid arthritis. Orotic acid is combined with PRPP to form UMP, uridine monophosphate. Orotic acid plus PRPP. It forms UMP. Uridine monophosphate. If the enzyme here UMP synthase is defective, orotic acid accumulates causing orotic aciduria, crystals in urine, failure to thrive. And failure to thrive. Orotic aciduria cause crystals in urine and failure to thrive. This pathway flows from UMP to UDP. This pathway flows from UMP to UDP. To turn RNA nucleotides UDP into DNA nucleotides, RNA nucleotide UDP to turn into DNA nucleotide DUDP. DUDP. We need an enzyme. That enzyme name is ribonucleotide reductase. That enzyme is ribonucleotide reductase. Drug block. Which drug block this process? Hydroxyurea inhibits this enzyme. This is used in sickle cell disease. Hydroxyurea. Hydroxyurea inhibits this enzyme. This is used in sickle cell disease. Now, in thymidine making, DUDP becomes DUMP. The final step is turning DUMP into DTMP. DUDP DUDP becomes DUMP. The final step is turning DUMP into DTMP. In final step, DUMP turn into DTMP. The key enzyme here is thymidylate synthase. Thymidylate synthase. It adds a methyl group to uracil to make thymine. It adds methyl group to uracil to make thymine. Drug block 5-fluorouracil 5-FU is a suicide inhibitor that mimics uracil and permanently jams this enzyme. Five fluorouracil is a suicide inhibitor that mimics uracil and permanently jams this enzyme. It mimics uracils. Permanently jam this enzyme. Now we will discuss about the folate cycle connection. You cannot make thymine DTMP without folate vitamin B9. We cannot make thymine, so here DTMP is thymine. We cannot make thymine without folate. That is vitamin B9. Six mercaptopurine azathioprine. They block de novo purine synthesis. Hydroxyurea. Hydroxyurea blocks the conversion of all RNA nucleotides to DNA nucleotides. Ribonucleotide reductase. all RNA nucleotides. Hydroxyurea blocks all RNA nucleotides. So it was summary of drug classes: leflunomide, methotrexate, trimethoprim, pyrimethamine, 5-fluorouracil, 6-mercaptopurine, azathioprine, hydroxyurea. So if you understand these lectures, kindly do subscribe to us for further in detail lectures. Thank you so much. Now we will discuss about purine synthesis. Unlike pyrimidines, the purine ring, adenine and guanine is built directly onto the sugar ribose. Directly built on the ribose. The starting point here is ribose 5-phosphate. It is converted to PRPP, phosphoribosyl pyrophosphate, by PRPP synthetase. Ribose 5P is converted to PRPP with the help of enzyme PRPP synthetase PR. This enzyme name is pyrophosphoribosyl pyrophosphate synthetase. The ingredients to build the complex double ring of purine: you need aspartate, glycine, and glutamine. To build the complex double ring of purine, we need aspartate, glycine, and glutamine. So there is a mnemonic gag: glycine, aspartate, and glutamine. 6-mercaptopurine drug and its prodrug azathioprine act as fake purines. They inhibit the conversion of PRPP into parent purine IMP. PRPP is converted into IMP, that is parent nucleotide. The drug's 6-MP and its prodrug azathioprine. These two drugs block this pathway. They act as fake purines. They inhibit the conversion of PRPP into the parent purine IMP. IMP is inosine monophosphate. It is parent nucleotide. It split further into become either AMP or GMP. IMP further split into GMP or AMP. Mycophenolate and ribavirin inhibit the enzyme IMP dehydrogenase. Mycophenolate and ribavirin inhibit the enzyme IMP dehydrogenase. IMP dehydrogenase, especially stopping the production of GMP. So drugs mycophenolate and ribavirin, they stop IMP and stop the production of GMP. Discuss about the mechanism to turn DMP into DTMP, to turn DUMP into DTMP. We need an enzyme for that methyl group. That donor is N5 N10 methylene. THF, a form of folate. It is a form of folate. After donating the methyl group, folate become DHF. After donating methyl group, the folate becomes DHF, dihydrofolate. It must be recycled back to THF, tetrahydrofolate. The enzyme dihydrofolate reductase is used again. Dihydrofolate reductase is used again. The result, the cell runs out of active folate, cannot make DTMP and DNA synthesis stop. Cell cannot make DTMP and DNA synthesis stop. This was pyrimidine synthesis. Now we will discuss Now we will study about a summary of drug classes. Leflunomide. Leflunomide blocks pyrimidine synthesis by in Orotic acid step. Block pyrimidine synthesis in Orotic acid step. Methotrexate TMP pyrimethamine block folate recycling stops thymidine. Methotrexate TMP pyrimethamine block folate cycling. 5-FU 5-fluorouracil directly blocks thymidine synthase